So when I was asked to do this inaugural lecture, I Googled inaugural, as one does, and I discovered that after the lecture, the candidate is presented with a cap, such as when you graduate, and a ring. So I'm looking forward to that. Some kind people also told me that I could do whatever I liked with this talk, but I should try to please everyone. And we know how that usually goes. So, and I must do it all in 2 hours. I'm sorry, that was a joke. Okay, so this has actually been covered. I was going to talk about population movements later on, and this is just mine. So here's the Eastern Cape, Queenstown, Windhoek, back to Grahamstown, Rhodes University, undergrad, where I actually started studying linguistics and then saw the light and went to science. And then postgrad in UCT, postdoc overseas in New Jersey, and then back to Stellenbosch all those years ago. So over the years, I've actually abandoned a number of promising careers. Firstly, in ballet. Secondly, in nature conservation. But I decided it would be very hard to get a job, and the pay wasn't that great. Then dragon boating, that's no pay at all. So I have stuck with science nerd. Now, there have been a couple of raised eyebrows about the title, and there was a book written in the 50s by Dubos and Dubos called The White Plague, Tuberculosis, Man and Society, and it's a classic in the field, so TB researchers know that term. But just to make sure, I decided to Google that as well and came up with this definition, which was the one to tuberculosis, which apparently makes some people appear pale. It was very serious in the 19th century. Hundreds of people died, thousands of people died, and it was often thought to be due to witchcraft or even punishment by God. But then on the same webpage, I discovered another reference, another definition to the British colonization of parts of the world, which I wasn't expecting. And I'm not quite sure why the British are singled out, because the rest of Europe did their bit as well. And then worst of all, the third definition is a novel about a bioengineered plague that kills females. So we can see that the Women's Task Force has a lot of work to do. But anyway, if you can see here what has been covered so far, basically there's religion, there's racism, there's sexism, and this is the world of genetics. We touch on everything. So I don't need to convince anyone here about the importance of TB. My work covers TB and genetics. TB is still one of the worst infectious diseases in the world. 10 million people, new incidence cases arise every year. 1 million of them have HIV, 500,000 have multi-drug resistant TB, which is very frightening. And 1.5 people actually die of TB every year, even though it's a curable disease. Then genetics is important because in every cell there are chromosomes which have genes which are translated into proteins and then control whatever happens in that cell. Genes are inherited, and the definition of heredity is what sets the parents of a teenager wondering about each other. Now, nature versus nurture is an ongoing debate. There are very few things that are solely due to environment, but there are some. For example, when I say religion here, I mean what religion do you practice? Hindu, Buddhism, etc. But there are a number of psychological components of life that are largely controlled by genetics. Also physiological. Lipoprotein A, for example, is hugely genetically determined, and the estimate for TB is over 60% genetically determined. The evidence that we have for it is wide-ranging. It's naive populations that have a death rate that decreases rapidly, probably due to selection. an incident where babies were immunized with MTB, and the response was very varied, probably because of their genetics. Twins are more likely to be similar to each other in terms of TB if they are identical, showing the genetic component. And then in animal models, we can actually manipulate the genetics and see the effects. So in TB, the first thing that one needs is the bacterium. Secondly, the environment is very important. And here, the environmental factors are things that affect your immune system. So HIV would be the most serious, steroids, poor nutrition, drinking, smoking, etc. And then the host gene environment, or the heredity and susceptibility genes in the host. If all of these are the wrong sort, then you are likely to get clinical disease. But your genes are not a sentence. There's always a lot of environment to work with. It's more like, I'd like to say, a hand of cards that you are dealt, and then it's up to you to decide how to play them. So I've mentioned the last serious epidemic in Europe was during the Industrial Revolution, when at least 25% of deaths were ascribed to TB, and we're not sure what happened in sub-Saharan Africa. TB affected everyone, from kings, poets, peasants, of course. It was thought to run in families. For example, if you look at the Emersons were badly affected. All the people on this page have had TB. The Brontes too. The father only died at the age of 89, although he had TB most of his life. And he passed it on to his children, who weren't so lucky. So TB is a problem. We're looking at millions of deaths every day in the future. The vaccines that are available are not very effective. There are multi-drug resistant strains, as we've said, so that's a problem. And this is our motivation for looking more at the host capability of controlling the disease. Because 5 to 10% of people infected with TB develop the disease. But the important thing is 95% do not, and this is because of their genes, we feel. So my group does research on host genetic susceptibility to TB, and as has been mentioned, we use a variety of genetic tools, and I will touch on some of these, but not all. So how does one catch TB? You catch it from a TB patient, usually by inhalation. And there are a couple of scenarios. One can develop acute TB. This is terribly simplified, by the way. The tuberculosis bacterium can be killed, and there are various genes involved in that. Or the most common scenario is that there's containment of the bacteria in a granuloma, and that the patient can stay with granulomas in their lungs for the rest of their lives and never actually have TB disease. If the immune system is affected such that there's reactivation, the bacteria break out of the granuloma and you turn into a TB patient. So we've done association studies on all the genes here, and the only one I'm going to mention is TLR, or toll-like receptors. They are receptors that bind and recognize specific molecules on the tuberculosis bacterium. And we found an association with TLR8, but it went in different directions in males and females. So why would this be? TLR8 that we were looking at is on the X chromosome. The association that we found was also found in Indonesia and Russia, and they didn't actually report on their females, but it seemed that it went in the same direction. And the interesting thing about toll-like receptors is that some of the genes that modern humans picked up when they left Africa and met the Neanderthals in Europe have been maintained. And people recently, this wasn't my work, but they looked at 3 of these archaic genes and found that they'd been strongly selected for. And when they looked at the function, they were found to boost the innate immune system by regulating the expression of toll-like receptors. So if that sort of thing is kept, you know it's important. There's a male bias in many infectious diseases, which means that males have a higher, and this is in blue, a higher incidence of those diseases and intensity. In some cases, females have a higher severity of a disease, but of course you have to have it in the first place before that's a problem. In the disease we're looking at, MTB, there's a male bias across the board. Why would this be? It's down to the X chromosome. Females are not invisible to bacteria, but we have 2 X chromosomes. So if there's a problem with one of the X chromosomes, there's a backup. Males have 1 copy, and therefore there's no backup. And the X chromosome contains mainly immunomodulatory genes. So females have been found to have a stronger immune system than males. They have higher white cell counts. They have more interferon gamma, more IP10, a number of things like that. Interestingly, in the genome-wide association studies that have been done so far, often the X chromosome results have been ignored. And the reason for this is what I've said before. Females have 2, males have 1. Often the female X chromosome has some inactivation, and all this complicates the statistical analysis, so that the software for analyzing the X is not very well developed, and it's easier to just ignore it. Nowadays, the new chips have better coverage on the X chromosome, and better software is being developed, so we are hoping to find some really interesting results on the X chromosome. We have looked at studies in buffalo who have TB as well. It's bovine TB. The origin is actually from humans who passed it on to their livestock, and then the European cattle came to Africa and passed it on to the wildlife. So we did some novel SNP discovery, and these short reads had to be mapped to the cow genome. And this is because the buffalo genome had not yet been sequenced. But the reason I want to show you this specifically is because, again, it's an indication of the importance of the toll-like receptors, because a growth inhibition assay in whole blood implicated toll-like receptor 6. So these are really important molecules. And then hot off the press, I want to mention that we have just completed the first whole genome sequencing of the African buffalo, and that will be published very soon. So on every human cell, we have HLA or human leukocyte antigen variants, and these are what's used for tissue typing. The variation is enormous between individuals and between populations, and the reason for this, for example here, if you look at the different colors, it's different frequencies of 2 specific HLA molecules, and there are hundreds of them. As people moved out of Africa, they encountered new pathogens and new climate, new food, and they selected for specific variants, which then led to different groups, which we now call ethnic groups. The other function of HLA is to actually present bits of protein called antigens from various bacteria to the immune system, so that the immune system is then reacts in the correct way. So as humans moved out of Africa, they also took their pathogens with them, and this indicates the movement of MTB out of Africa, and different strains of the bacterium also developed. When the population in various centers grew, and we're talking about this time period here after 1000 AD, the TB bacterium did very well in these large, dense population centers. Each dot here, each dot is 1 million people. And then the strain started moving with the people again, and everybody came past the Cape, which was known as the Tavern of the Seas, and they all dropped their strains there. So we have an extraordinary variety of TB strains at the Cape. So this study looked at 300 TB cases, which we then typed for their HLA, and we also genotyped their tuberculosis strains. And when we looked at logistic regression models, which is a statistical way of looking at the data to find patterns, we found a favored combination of many of the pathogen genotypes and specific genotypes of the host. And this is because of co-adaptation between the host and the pathogen. There's a constant arms race. What we should also notice from this is that vaccination with a specific strain is not going to be the best thing necessarily for all populations, but it's really a one-size-fits-all at the moment. One of the biggest studies we did was a linkage study, and normally one looks for enormous families in genetics. But these days in the suburbs, everyone has 2.3 children, so we rather went with looking for a large number of families. We did try to find families with large numbers of children and people with high exposure to mycobacteria. And then we measured the tuberculin skin test, which is you inject PPD into the arm, and if there's a reaction, it's a signal that the person is actually infected with TB, even though they may not be ill. So the chromosomes are arranged along here, 1 to 22, and we found a linkage peak at chromosome 11 that determines whether one has a positive tuberculin skin test or not. Another locus or area on chromosome 5 determines how big that reaction is. And we felt that this was really important because especially the people who do not have a TST reaction at all, even though they're in a high TB environment, are likely or possible that they are resistant to TB. But there was a lot of resistance to this idea. Finally, the penny dropped, and the NIH put out requests for proposals, and they have now funded us, and we're about to start on a study to look into this phenomenon, which is probably going to be really interesting, to me anyway. In the meanwhile, we have validated the study in a population in Paris that has different genetics, obviously, from the Cape, and a much lower incidence. So completely different environment and genetics, and we come up with the same thing on chromosome 11. And even more exciting is that we looked at the locus or the genetic area that controls the secretion of a cytokine called TNF, which controls the formation of granulomas. And this overlaps very nicely with this TST1 area, and therefore may have something to do with the resistance to TB. Okay, very briefly, we did a genome-wide association study. We found 2 new loci. We replicated one that had been found before, but the most interesting thing that came out of this study was that there's a positive correlation between African SAN ancestry and TB susceptibility. And we'd always wanted to do admixture mapping. Admixture mapping uses an admixed population from 2 or more genetically different parent populations, where the incidence of the disease that you're interested in is also different in those parent populations. And there have been a large number of successes with various diseases where that applies in African Americans. They have a very nice 2-way admixture between West Africa and Europeans, and things worked out very nicely for them. So we wanted to do the same. So the first question is the incidence of TB different between, say, blacks and whites? So this is a map from 1900. Firstly, it shows you where TB came into the country. And secondly, if you look at the rates, this is actually death rates because it's pre-antibiotic era. The Europeans that were living in South Africa had a rate of 1.5 per 1,000. The ones that back home in England and Wales had almost exactly the same rate. So this seems to be common to the population and totally independent of the environment. The Kanet and the Bantu in this area had a rate 5 times higher. So we decided to go ahead with this admixture mapping. Our participants were from the South African coloured population, and I know some people don't like the term, so I apologise if you don't. We're simply using it because it's a census term and our population self-identified as such. We genotyped 1,000 people. We looked at a chip that has 500,000 single nucleotide polymorphisms across the genome, which of course then generates 500 million data points. This is what a chip looks like, just in case you were wondering. It's a small chip, and on it you print 1 million or more DNA probes, and every time you move DNA from a participant in your experiment over it, you generate 1 million pieces of data. So this very rapidly leads to a large number of data points, which is known as a very large data set, but colloquially we just call it big data. And this has completely transformed genetics in the last 10 years. It's a completely different subject, really. And this big data can be extremely startling to students. This is actually me. In our case, we had the added complexity of the population genetics. We did not know what the genetics of the population was. There was some historical data on the colored population, but no one had published any genetic data to answer these questions of who are we and where are we from. So one of the parent populations is the Khoisan. This is the current distribution, but if you know what is in this area, you'll know that no one would choose to live there if they could live here. And the original distribution was throughout South Africa, but these people were displaced by migration, etc. So they were originally found then throughout southern Africa. And then in about 3000 AD, roughly, there was a movement that started in the Cameroon area called the Bantu expansion, which came across the continent, down the east coast, and last of all in the tip of southern Africa. In 1652, the Dutch East India Company started their refreshment station. This point here actually is Mapungubwe, almost, and this migration had reached there in about 1000 AD, just to give you the time scale. A year after the Dutch settled here, they started importing slaves. The slaves in the Cape came from all around the Indian Ocean, from East Africa, Madagascar, Sri Lanka, Bengal, and Java and Sumatra. These were often political prisoners, actually, from the other Dutch colonies. So we looked at the population structure via genetics because it affects all of our genetic studies. We selected what we thought were probable ancestral populations from various international genetic projects, and we then analyzed these by programs such as structure and principal component analysis. A structure plot looks like this, where each vertical line is an individual. So an individual here, for example, has some red, which is from the European population, then some blue, which is from the Asian component, input from green, which is San, and then some from the tan color, which is African. In this case, we used Yoruba as a proxy for African ancestry. And what you can do with a set of samples like this is 1,000 people. You can actually average them, but we know that there's no such thing as an average person, but it's just to get numbers. So 2 studies were done as more and more SAN data became available. And in the end, we can say that this is very approximately what the South African colored population is made up of. It's approximately a third Khoisan, just a bit less than that Bantu-speaking African, about 20% European, and about 20% Asian, East and South Asian. But there is enormous variation between individuals. Here, those 1,000 people are sorted according to the ascending contribution of Khoisan, or African, or European, or Indian. So you can see whichever way you arrange people, you can see this tremendous variation. We did a collaborative study looking at gender specificity and found a very big gender bias, which is common in colonial situations. So here, pink is female, blue is male, and you can see that the female input is very, the female input of the African parentage is very high. but European female input is very low. The paternal input is African and European. And then if we blow up this African component into Khoisan specific and Bantu specific, and the rest cannot actually be distinguished, we can see that the Khoisan specific maternal input is tremendous, whereas the male input is not so high. And to understand that, one needs to look at what we do know about history. This is a Khoisan clan moving with their cattle. But as we all know, the land was taken away from them. They lost their cattle. Smallpox totally decimated the population. There were entire groups that were just totally wiped out. So the people that were left then had no land and became laborers on farms. So the men tended to be these bonded laborers in the colonial society. And women were more integrated into the society, often by marriage. The other parental ancestors are the non-Khoisan Africans, largely in this case the Xhosa, and they had already in fact admixed with Khoisan to a certain extent as they moved into southern Africa. Then of course there were the Europeans, Dutch, German, British and French, who were the landowners. There were a large number of male settlers, especially at the beginning, but a drastic shortage of women. So a lot of these landowners would actually import wives. Sometimes orphans came out from the Netherlands and France, or they would marry Khoi women. And if you look at the slaves that came into South Africa, into the Cape area, 26% of the total came from Bengal. It was also a Dutch colony, so it was convenient for them. Again, mainly males, very few females, but often the female slaves were actually manumitted or freed and then often married their owner. This is in fact a roll of names in the slave lodge in Cape Town, and the names are Van Batavia, Van Bengal, Van der Kaap, if people were born in the Cape. This was a very successful landowner. He was actually born in Mauritius before he came to the Cape, and his mother was a freed Indian slave. His name was Simon van der Stel, and he gave his name to the university. So these are just scenes of the early Cape, slaves doing fishing, selling goods, laundry. But in 1834, under the British rule, there was emancipation. Slaves were freed. The ones that did not want to stay with their previous owners often flocked to the mission stations, and they were then joined by the landless Khoi. And this is just my own very small family contribution to the abolition of slavery. This is my paternal grandfather, W.G. Hoal. He was the Secretary for Justice in 1939, and he was asked to clean up the Cape Statute Book. So he collected about 1,000 ordinances that were totally out of date, including one from 1683, providing for the baptism of slave children. There was another one legislating the size of the parasol that a lady was allowed to wear or carry in Adley Street. That was taken off as well. Okay, so back to TB susceptibility. A PhD student developed some ancestry informative markers, and we went back to our 1,000 TB cases, and we did another 500 controls because the previous results had been done with very few controls. And so with these much better numbers, we then did another logistic regression study and confirmed that SAN ancestry is a risk factor for TB. It's not predictive, for example, the controls. So there's a highly statistically significant difference, but you can see that there's a large overlap as well. We continued doing admixture mapping, but we needed to do local ancestry inference to find out which parts of the chromosome came from which ancestor. And in 2013, the algorithms for this were really not good enough. But in 2014, a new algorithm came out, and we could do the local ancestry inference accurately and finally we could do admixture mapping. So these are all the chromosomes. I'm certainly not going to go into all of this. The results that we found then, we looked at regions in the cases that had excess San ancestry and regions in the cases that had excess African ancestry. And we are hypothesizing that these may contain susceptibility genes. And we will be looking much more closely into these regions. So to end off then, I'm just going to talk a bit about population movement, specifically in Southern Africa. So the origin of this first biggest population movement should actually start way down in Southern Africa, because we think that's where the origin of mankind was. And modern humans then moved out along the coast of the Indian Ocean. They moved into Asia, they moved into Europe, and finally into the Americas. In the Cape, and this is way before there were any settlers, there were visitors, but no settlers, there were a number of Khoi groups and their cattle in sort of mutually agreed regions. They had very well developed trade routes that went into the interior. And if you know the map, you can recognize the N1 and the N2, which were really, they were trade routes long before they were tarred. And another route went up as far as the Orange River. So these went through the regions of the various Khoi groups, and they had an extremely good bush telegraph. They would know within a couple of days what was going on in Delagoa Bay. So the Dutch in Cape Town heard stories of the interior, of enormous lakes and people who lived in stone houses and people who mined gold. And first they got very excited. After a while they got really very skeptical, because they never found these things. Probably because they mounted expeditions into the north, always in February. That seemed to be the favorite month. And so most of them died and didn't find anything. But all of those stories were in fact true. This is the lake. The Orange River used to flood regularly. There were no dams. So every year there would be a large lake forming there, which would stay for about 4 months. So the Khoi in that area did not have to move anywhere with their cattle. They could live there permanently. And we've talked about the trade routes from the Cape to the Orange River, from the Cape across to the now Transkei. But there was also a very active trading post in Delagoa Bay, the Portuguese and the Arabs. And you can see it's a very short hop to the headwaters of the Vaal River. And once you have a river, you have a very good trade route that you can walk along or go on horseback. And when the explorers made it to the Orange River, they found all sorts of trade goods that really could only have come, they didn't actually realize it half the time, but now we know they could only have come from Asia. There were beads specific to India, there were plants such as willow, even dacha that everyone was smoking at the time. That is not indigenous, it comes from Asia. So I thought maybe we should rewrite a bit of Latin. This famous quote that says, "From Africa there's always something new," from Pliny the Elder, who actually got it from Aristotle. Perhaps we should change it to, "There's always something new coming into Africa, and not always for the good." So the trading that was going on here was actually very destabilizing for the societies. And the knock-on effects went all the way up to the Orange River, and then renegades started arriving in the area, and the entire social fabric was a complete mess. A similar thing happened here in Delagoa Bay. The traders here sort of rekindled rivalries and hostilities between these tribes, and this entire society was also totally destabilized. In the early 1800s, there was also a very severe drought in this area, and then a king called Shaka. And this led to the Mfecane, which was a disaster for the country. Hundreds of thousands of people died. They either died during the war or they starved to death. The MPs often implemented the scorched earth policy, so you either starved or you moved. So there were these enormous population movements, even into neighbouring countries, right down to the borders of the Cape Colony. And I was thinking at the time when I was reading the history, really, I stopped watching Game of Thrones, because if you read South African history, it's got everything and more. So the point here is that any groups that were stable before about 1800, we don't even have any records for those areas. And the last example I'll show you is of population dynamics is the Wild Coast, which is wild. It's estimated there are about 2,200 shipwrecks along the coast of South Africa, which averages out to about 1 per kilometre, and they're going to be disproportionately clustered in the Wild Coast because of the currents and the winds and the reefs. And every time there were castaways that actually made it to the beach, they would always find other castaways from previous wrecks. So this was a really common thing. And often the castaways had been taken into the local tribes and looked after and integrated. There was a group called the Abalungu, which means the white people. They came from white male castaways who then married local women and started their own clan. There was a 7-year-old English girl who was shipwrecked here and grew up to become the great wife of the chief of the Amachomani, and her descendants are still found in the royal families here. So the point is that everyone in South Africa is a lot more admixed than they perhaps think. And to do population genetics in a situation like this is quite challenging. And we need to just be aware of what we're doing. So to conclude then, we look at these human gene variants that are involved with TB susceptibility, because we think that they will give good clues for better therapies. And if you're working with a population that no one else has worked with before, you may get novel ones. So we've gone through the dynamic history with populations moving and mixing over hundreds of years. We also have a variety of TB strains, and this gives us many permutations and combinations between the human and the bacteria. And I personally, and it's not only me, feels that this interplay between the bacterial and the host genes is really the key to deciding whether a specific individual is going to get ill or not and could control the epidemic. And these are just interesting history books. Thank you for your attention.